Alright, we're going to continue with part 2 of our cell lab on cell and mitosis. So we're picking up with this slide here on parts of a chromosome. And click here. Looking at objective number 4 in your lab packet. So this is on page 8 in your lab packet that you purchased on in the bookstore. Looking at the parts of a chromosome. So this entire structure here is a chromosome, the whole thing. And basically what a chromosome is, it's just like chromatin in that it's DNA. So chromatin, we mentioned last time, is DNA. It's found in the nucleus. But chromatin is very loosely arranged. It looks like a pile of spaghetti. It's basically DNA nucleotides that are just loosely arranged and in the nucleus. Then this DNA can tightly coil to form a spring-like structure, which is called a chromosome. So chromatin and a chromosome are both DNA, but a chromosome is more tightly wound. So here we can see how it's tightly wound into this X-like structure. One chromosome would just be one side of this is one chromosome, and the other side is the other chromosome. It actually goes like this. It comes down and then like that, and the other one comes down and like that. So together it's a chromosome. Each side of this chromosome, each side of the X, so this side is called a sister chromatid. The other side is a sister chromatid. So when they use the word sister, it basically means they're related. Both sides of this chromosome are related in terms of the DNA molecules that they are. So they have similar information. One is a complementary strand to the other. So again, it's a sister chromatid, one half of this chromosome. So the entire structure is a chromosome. What holds these sister chromatids together is what's called a centromere. That's what the red dot is showing here on this diagram. That's a centromere, and it holds those sister chromatids together, and it is also what the sister chromatids, what the chromosome is going to attach to in mitosis when the cell gets ready to divide and split this chromosome into two parts. If we look at a map of our cells, we would get something like this that's called a karyotype. And a karyotype shows a map of our chromosomes. So every body cell, whether it's a skin cell, muscle cell, any cell in the body, it's going to contain 23 pairs of chromosomes. So this is chromosome number 1, chromosome number 2, chromosome number 3. There's a pair. One came from our mother in the egg that produced us, and the other one came from our father in the sperm that produced us. So we have 2 of each chromosome. So 23 pairs for a total of 46. So here we can see chromosomes 1 through 22. And then these last chromosomes here, this pair, are the sex chromosomes, and that determines whether you're going to be male or female. So here we have 2 X chromosomes. This would be indicating this came from a female. This one here, the sex chromosomes are X and Y. This came from a male. So in looking at this, we can see we have a female karyotype on the left and a male karyotype on the right. So each of these bands on these chromosomes represents a gene, and it codes for specific information. And some things that are coded for by our genes would be like hair color, eye color, certain genetic characteristics that tend to run in families. They're carried by genes on chromosomes. Sometimes we have more than one gene on more than one chromosome that would determine, for example, eye color, hair color. But in general, these chromosomes carry genes which code for specific proteins that determine structure and function of the body. So this is objective number 5 that asks you to state the number of chromosomes in a body cell. So you would say 23 pairs or 46 total. Objective number 6 asks you to identify the stages in the life cycle of a cell. Well, the main primary stage that our cells spend most of their time in is called interphase. And here we can see a group of cells. These are whitefish blastula cells, which are early growing fish cells, and these are all in what we call interphase. Here we can see the plasma membrane of a cell, of a fish blastula, and then here's the nucleus. But we can see there's a nuclear membrane. You can see how it's very dark and distinct on the edge of this nucleus. We can even see a nucleolus in there. So this would be a cell in interphase, very characteristic of interphase. Again, dark membrane. You can see a clear membrane around this nucleus, and we can see a nucleolus in there. That's indicative of interphase, and that's the non-dividing phase of a cell cycle. When a cell gets ready to divide, there's 2 processes that occur. The first is mitosis, and that's followed by cytokinesis. If we look at this pie chart here, we can see that most of the life cycle of a cell is an interphase. More than 3/4 of this life cycle here is spent in interphase. Then it enters into a brief phase of mitosis, followed by cytokinesis, and then back to interphase again. So the purpose of cell division is to replace cells or to increase the number of cells during growth or repair. So we'll look at each phase of mitosis and be able to identify it based on the cell characteristics. So when you talk about mitosis, the clear definition that you need to know when you hear the word mitosis, it's division of the nucleus. So here we have a cell with a visible nucleus, and by going through the stages of mitosis, we can see that nuclear membrane breaks down, the chromosomes are scattered throughout the cell, they line up in the middle, they're pulled apart, the cell pinches in the center, the nuclear membrane reappears, and you have 2 new cells from the original parent cell. That's mitosis. Mitosis is essentially a copy machine where we have 1 cell in the beginning, and at the end of mitosis we have 2 cells. Again, the definition is division of the nucleus. Highlight, underline the word nucleus. Because the other step in cell division is cytokinesis, and that's at the end of mitosis. This is defined as the division of the cytoplasm. Again, underline, highlight the word cytoplasm. Here we can see this cell is near the end of mitosis, and the cytoplasm, the plasma membrane, is pinching in on the edges. And that's going to continue to happen until this pinches in to form 2 completely new cells. And that's cytokinesis, again, dividing that cytoplasm into 2 new cells. So when we get to the phases of mitosis then, when a cell is at the end of interphase and there's a signal for the cell to divide into 2, the first step of mitosis is called prophase. And we can tell that a cell is in prophase because the nuclear membrane disappears. Here we don't see a visible membrane, and we also see chromosomes start to appear. Here we can see strands of chromosomes showing up here. Here we can see thin strands of chromosomes showing up. So the DNA is no longer in the form of chromatin, where it's loosely arranged more like a plate of spaghetti. Now they're coiling and condensing into these spring-like structures that we know as chromosomes. So this is a cell in prophase. Metaphase is when those chromosomes are pushed to the center and form a straight line in the center of the cell. So when you hear the word metaphase, think meta, middle. Cells are lined up in the middle of the cell. So here we can see there's a nice straight line of chromosomes pushed to the center by those structures we talked about in the first part of our lab discussion, the centrioles. And I'll show a video animation at the end of this presentation which will show you how that happens. But here they're lined up in the middle of the cell. That is characteristic of metaphase. Now these structures you see here, these are not nuclei. These are actually centrioles that are secreting a spindle that push the chromosomes to the center. The cell with plasma membrane is here. This is the whole cell here. And these are centrioles here that are pushing these chromosomes to the center. Then the next phase starts, and that's anaphase. These centrioles that push these chromosomes to the center have that cage-like spindle fiber. Now that fiber shortens and starts to pull the chromosomes apart. Remember we talked about the sister chromatids? These sister chromatids are pulled apart from one another now, one half going to one side of the cell, the other half going to the other side of the cell. So when you hear the word anaphase, think apart. Anaphase, apart. They both start with an A. Here we can see a space between those sister chromatids as they're being pulled apart from one another. The last phase is telophase. Telophase is where we can clearly see cytokinesis is well underway, and this membrane is starting to pinch in the center. And we can see those sister chromatids that were pulled apart. They are on either side of this cell. Here we can see again, here's this one half of the sister chromatid, here's the other half. Those chromosomes were pulled apart and the cell membrane starts to pinch in. So when you see a cell in telophase, you're going to see this characteristic figure 8 appearance. So look for that figure 8 appearance to indicate telophase. Here again we can see a figure 8 appearance of indicating telophase. Now in your lab packet on page 9 I have some pictures there for you to reference to help you identify the different cells in their phases of mitosis. If you look at page 9 on the top of page 9 on the right hand side, what you want to write in the square there is interphase. That's a cell in interphase. And the arrow is drawing your attention to the characteristic feature of interphase, and that is there's a visible nuclear membrane. So I would write that next to that arrow there, visible nuclear membrane. The second picture in the middle on page 9, the second one down, is prophase. So you'd write prophase in the box. And next to the arrow, we can see visible chromosomes are showing. So you can start to see visible chromosomes in that cell. The third picture at the bottom of page 9, that's a cell in metaphase. So you can write metaphase in the box, and the arrow is pointing to the cells lining up in the middle. So you can write that by the arrow. Not cells, sorry, chromosomes. Chromosomes are lined up in the middle. On page 10, you can write in the box that's anaphase, and the arrow is showing chromosomes being pulled apart. Again, apart being the key word with anaphase. And the last square on page 10, the bottom square, that picture, that is telophase. And the arrow is drawing your attention to that figure 8 appearance, where the cytoplasm is pinching inward, getting ready to separate those new cells. So again, it's telophase, and the cytoplasm is pinching inward, or you can write it's giving a figure 8 appearance. On page 11, there's a couple of different cells with letters by them, and you're going to identify those. So on the bottom of page 11, you'll see I have the letters there, and there's space next to each letter to write the phase that you think that cell is in. What I would do now is pause this presentation and take some time to see if you can figure out what each cell, what phase they're in, and then I'll go over the answers when you're done. Letter A is in anaphase. Letter B is metaphase. They're lined up in the middle. Letter C is telophase. You can see that characteristic figure 8 appearance. Letter D is interphase. Can you see that dark nuclear membrane around the edge? Letter E is anaphase, because the chromosomes again are being pulled apart just like they are in letter A. And letter F would be prophase. We don't see a visible nuclear membrane there, but we can see little bits of stranded chromosomes, and that's characteristic of prophase. So that concludes the lab portion.